Hello my dear students. We will continue the questions of biology. Question 2, number C. If the sequence of nitrogenous bases in DNA strand is as follows. These act as a single strand of DNA. G is bold and underlined. Number 1, calculate the percentage of the adenine in the space of DNA double helix. The question produce one strand of DNA and need the percentage of the adenine. Adenine is the A. To calculate the number of adenine in the double helix or the two strands, we can calculate the A and the T in one strand. That means the number of A in the this strand is 1, 2, 3, 4. But the number of A on the other strand which complemented on this strand, that the number of A facing or paired with the T. So we can calculate the number of A and T in the single strand. The number of A and T, 1, 2, 3, 4, 5, 6, 7, 8, 9. That means 9 adenine are located on the double helix. 9 adenine, and the number or the total number of the nucleotides in the double helix is the 30. So the percentage of the adenine is the 9 over 30 times 100. That's will produce 30% of adenine, and also 30% in thymine because A equal T. But the G or the guanine is the number of A minus 50 or 50 minus A that produce G or C. So G will be 20 and C also 20. But the question asked about the percentage of adenine, and the percentage of adenine is 30. Number 2. The mutation occur replace the nucleotide G in this strand, which is colored by red color and underlined, but no change in the type of the amino acids. We understand that if one or more than one nucleotide removed or replaced or changed, that may change the amino acid. And the change of amino acids leads to change the protein produced from the translation of the mRNA that formed from this part of DNA. If the protein is changed, that means that change enzyme, change the character, leads to mutation, which is called the G mutation. But the question asked about that the change takes place in G, but no change in the type of amino acids. That means if the G is changed, that means that it changes the codon or the second codon only. The second codon, GCG. In the case of the mRNA, the codon will be CGC. But what about if the G is changed and no change takes place in the amino acids? That takes place because the amino acid may containing more than one codon. So if the amino acid containing many codons, that means the change of one codon or one nucleotide that will change the codon but not change the amino acids. For example, the arginine in this case, codon is CGC and CGA and also CGG. So the amino acids, if the nucleotide change, that may may change the amino acid, but also may not change the amino acid because each amino acid has more than one codon. But if the change of the nucleotide or the sequence of nucleotide change the amino acids, that called the gene mutation. But if no change in the amino acids, that means no change in the character or no mutation takes place. The next question. Study the diagram which represent the vertical sector of the human tests, and then answer the following question. That's act as a part or TS in the seminiferous tubules of the human tests. Indicate the function of the structure number 2 and number 3. Number 2 is the interstitial cells. The interstitial cells, that's a group of cells that located between the seminiferous tubules. The interstitial cells are responsible for the secretion of the male hormones or androgens. Androgens as a testosterone and androsterone, that responsible for the appearance of the male secondary sexual character. But the letter number 3 is the Sertoli cells. Sertoli cells are responsible for the secretion of fluid or nutritive substance to feed the sperms inside the testis, and also the Sertoli cells have an immunization function that to protect the sperms against any infection. Number 2, explain the chromosomal number of the structures 2, 3, and 4. Number 2 is interstitial cells. Number 3, Sertoli cells. Number 4 is cells which is called the spermatogonia. The spermatogonia, in this case 2N, and also the interstitial cells and the Sertoli cells 2N, each carry 2N or carry 46 chromosome. So all these structures or these cells containing 2N chromosomes, and they produced from the mitosis division. The third part, to draw the sperm. The drawing of the sperm is very simple, because we'll draw the head of the sperm containing a nucleus containing the genetic material that transfer from the father to the offspring, and containing a chromosome that responsible for the secretion of hyaluronase enzyme, containing the neck region with the centrioles which help the zygote to divide, and also the midpiece containing the mitochondria that responsible for the secretion of the energy that helps the sperm to swim to reach the ovum for fertilization. And the last part is the tail or the caudal piece that helps the sperm to swim to reach the ovum. The next question, explain how can we get each of the following. Fruits devoid of seeds or seedless fruit. Number B, frogs without fertilization of the ovum. To produce fruit without seeds, that means the parthenocarpy will take place. That takes place if we spray the stigma of the flower with the extraction of the pollen grains that dissolved in water or ether, that stimulate the ovary to grow to change into fruit without seeds. That is the formation of fruit without seeds. But which is called the parthenocarpy, and the parthenocarpy is not act as a kind of reproduction, but just growth. The ovary grow to form or to change into fruit. Number B, frogs without fertilization. If the frog egg or if the frog ovum is exposed to chalk or to a certain chemical substance, or salts, or exposed to electric shocks that stimulates the ovum nucleus to divide mitotically to change into 2N. So the ovum become 2N similar to the zygote, and it grow to produce the offspring similar to the mother, because that act as a kind of asexual reproduction, which is called the parthenocarpy, which is the ability of the ovum to divide to produce complete individual without fertilization. The next question, how many spinal nerves connected to each of the following? Cervical region. Number 2, coccygeal region. We understand that the cervical vertebra are 8 vertebrae, but the number of the spinal nerves get out from the neck region is the 8. But the coccygeal region, the coccyx consists of 4 fused vertebra, which is called the sacrum part. So the coccygeal, which is called the tail or the coccygeal part. The coccygeal part containing or produce 1 pair of the spinal nerves. So the cervical region produce 8 pairs, but the coccygeal 1 pair only. The next question, give reason for each of the following. The organs of the human female genital system are firmly attached in the pelvic region with the elastic ligaments. The elastic ligaments is very important to help the female reproductive system to relax during the presence of the fetus inside the pregnancy, and that to facilitate the delivery that will take place by the action of a certain hormone, which is called the relaxin hormone, which help in the expansion of the pelvis during delivery, during the pregnancy state, that to facilitate the delivery. Number 2, the several thousands of the ribosomes are produced in per 1 hour or in 1 hour in the case of eukaryotes. We understand that on the DNA that we explain that on the DNA the presence of 600 copies of genes that are responsible for the formation of rRNA. So the presence of great number of genes or great number of copies of the genes that are responsible for the formation of rRNA that will accelerate the formation of the ribosomes. So the ribosomes are produced in many thousands per hour. The third part is the blood platelets are continuously renewed inside the body. We understand that the blood platelets are very important during the formation of blood clotting that protect the body against the bleeding and also protect the body against the hypotension. So the presence of the blood platelets is very important, and also the blood platelets survive for not more about 10 days. So we need the production of great number of blood platelets from the bone marrow, that because of the importance of the blood platelets during the formation of blood clotting, and also the blood platelets survive for not more 10 days only. So we need to produce blood platelets continuously. The next question. The presence of a special purified liquid in the artificial kidney. The path of the artificial kidney containing the fluid which is called the dialysis solution. The dialysis solution containing all the blood components or all the plasma components except the urea and the salts. So the plasma which passes through the selectively permeable tube inside the semi-nephros tubules, or the blood of the patient which containing a high concentration of urea and the other metabolic wastes that form the urine, that pass through the tube or semi-permeable tube or selectively permeable tube that located or immersed in the dialysis solution. So the high concentration of urea inside the tube that causing the passage of the urea from inside the tube where the presence of the blood in the dialysis solution, and also the salts and the other metabolic wastes. So the presence of the dialysis solution that helps the passage of the urine components from the patient blood through the dialysis solution that takes place by diffusion, and also the dialysis solution pass through the filters to remove the urine components from this dialysis solution or the solution or the fluid inside the kidney. So the continuous movement of the blood inside the selectively permeable tube inside the artificial kidney that causing the passage of the urine components as the urea, salts and the other metabolic wastes from the patient blood to the dialysis solution. So that's act as a dialysis process. The next question. Write the scientific term for each of the following. A type of mutation that's occurrence at the it's attributed to the environmental factors around the living organism. That means this mutation takes place without the interference of the human being. This kind of mutation is called the spontaneous mutation because it takes place by the action of the environmental factors. Number 2, the membrane with the fluid that protect the embryo against the shocks and the dryness. The membrane which containing the fluid, that is the amniotic membrane, and the liquid inside the amniotic membrane is the amnion. In the amniotic fluid that's surrounded by the amnion that protect the brain against that shock, because that act as a mechanical shock, and also that protects the fetus against dryness. The next part. A coenzyme receives the hydrogen resulting from the splitting of the water during the photosynthesis process. we understand that during the light reaction, the high concentration or the high amount of energy in the active chlorophyll is divided into two parts. Part of the energy is used in splitting H2O into hydrogen and oxygen. The other part change ADP into ATP. But the hydrogen carried on the NADP and change it into NADPH2, and the oxygen is evolved. The coenzyme that responsible for receiving the hydrogen in the light reaction is NADP or NADP, that located on the chloroplast. Number 4, the bacterial enzymes that attack the specific nucleotide sequence in DNA molecule and break it into useless pieces. The question asks about the enzyme that cuts the DNA into useless pieces in a certain parts, which is called the recognition site. The enzyme is called restriction enzyme, and the restriction enzyme, the action of the restriction enzyme is restricted on a certain part, which is called the recognition site, to cut the DNA into useless pieces. Therefore, the bacteria which containing the restriction enzyme containing another kind of enzymes that are called the modification enzymes that responsible for put the methyl group on the recognition site to protect its DNA against the action of the restriction enzyme inside the cell. The next question. Mention the site and the function of each of the following: Casparian strip, nucellus tissue. We understand that the Casparian strip is located in the endodermal cells, which act at the innermost part of the cortex in the root. The Casparian strip made of supreme. The Casparian strip responsible for the help the water to pass from the root hair to the xylem vessels, but prevent the passage of the water to the phloem by imbibition, which is not controlled by the cell, because the Casparian strip is made of supreme. The next structure is the nucellus. We understand the nucellus act as the nutritive tissue that surrounds the embryo sac. So the site is surrounding the embryo sac. The function is the act as a nutritive tissue that to feed the embryo sac of the flower. The next question. Compare between voluntary reflex and involuntary reflex. We understand that the reflex action takes place by two kinds of muscles. The first is the voluntary muscles, and the other is the involuntary muscles. So the if reflex action takes place by voluntary muscles, that's called the voluntary reflex action, which is takes place by the skeletal muscles or the muscles that attach to the skeletal system. But if the reflex action takes place by the involuntary muscles, that called the autonomic reflex or the involuntary reflex action. Number 2C, in one of the crossing, the offspring with the genetic structure or models G small, L small, G small, L small 50%, explain on the basis of the genetic type case represented by the genetic hybridization. We understand that the presence of G small, L small, G small, L small, that means the case is a complete linkage, because the two genes G small and L small are located on the same chromosome and inherited at the one unit. So in the case of G small, L small, G small, L small, the ratio of 50% takes place by the test cross of impure individual with the pure recessive individual. The impure individual will be G capital, L capital, G small, L small, and the other G small, L small, G small, L small. So the gametes of the recessive individual G small, L small, but the impure dominant individual G capital, L capital, and G small, L small. If the ovum of the G small, L small is fertilized with the sperm G capital, L capital, that will form G capital, L capital, G small, L small, that will be gray long. But in the other case, if the ovum G small, L small is fertilized with the sperm G small, L small, that will form G small, L small, G small, L small, that will be the recessive individual. So the ratio will be 1 to 1 or 50-50 and this case is a complete linkage. The next question. Correct the underlined words. Enterokinase enzyme catalyzed the breaking down of the protein into polypeptide inside the duodenum. We understand that the enzymes that are responsible for the hydrolyzing of the protein into polypeptide, two enzymes, pepsin inside the stomach and the trypsin inside the duodenum. The pepsin inside the stomach is secreted in the case of the pepsinogen is activated by the HCl inside the stomach. But the question asks about the enzymes that catalyzing the breaking down of the protein into polypeptide. The enzyme is trypsin, but the trypsinogen is not act as the right answer because the trypsin can't change the protein into polypeptide because the trypsinogen in inactive state. So the answer is the trypsin. The next part. The unit function of the skeletal muscle is known as the axon. The unit function of the skeletal muscle is called the motor unit. That's not act at the axon. The swelling structure of the mimosa leaflets act as a muscle during the movement. We understand that the pulvineus, which is a swollen structure as the primary rax, and the base of the secondary rax act as a joint, because when it loses the water, which act as a physiological support, and the water get inside the cavities that help the or causing the wrinkle of the pulvineus, so it act as a joint and help the leaf to or the rax to fall down. So we will change the muscle into joint. The next part, the pulling roots wilt and die if they don't meet the support during the twinning of movement. We understand that the pulling roots is not need the presence of support because the roots move around or twist around the soil particles to pull the pulps and keep them at a suitable distance from the ground. But in this case, the tendril needs the presence of support. If the tendril not found the support, that means the tendril will wilt and die. So the pulling roots is not right, but the right is the tendril. My dear students, that's the end of this part. Thank you very much. Goodbye and good luck.